Welcome to Efficiency Gains with Sequence Variant Analysis by Mass Spectrometry. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. The need to efficiently detect and identify variants of all types has grown rapidly. Our speakers today will talk about how modern mass spec and informatics can provide efficiency gains in sequence variant analysis. They will also discuss how the continual use of accurate mass data with informatics can inform the clone selection process and how LCMS can be incorporated into the clone selection process. If time allows, they will be available to answer your questions on best practices as well. Our first speaker is Sinjin Skilton, Senior Global Marketing Manager, Biologics at AB Science. Sinjin will provide a brief introduction on sequence variants in biologics and the challenges associated with sequence variant analysis. Sinjin, so as not to steal your thunder, I'm just going to hand the mic over to you and get this webinar underway.
Hi, Tamlin, and thank you very much for the introduction. And thank you, of course, to all of our listeners and attendees for attending this webinar. And welcome to the world of logical biologics. So today I'm going to provide a very brief introduction to the topic by highlighting some of the key developments that have made advanced work possible in recent years. So crucially, as is often the case with many of these developments, a number of factors have had to come together. So, of course, informatics plays a large part, the brute instrument capability, market forces that provide the commercial potential for this sort of thing, but perhaps most importantly, the scientific creativity to see better ways to bring together these tools. And I think that after my talk, you will see that in abundance in Jason's presentation in a moment. So we all know that cell lines and the expression of recombinant proteins is not a static thing, but very dynamic. And we all know that we can't really predict what's going to happen with different cell lines and different harvesting techniques or purification in terms of the sequence variants that appear. So what we really need to do is be able to look for them without any preconception in an unbiased manner. And a crucial issue for us in the vendor world is that the new sequences tend to be very low in abundance in many cases, and therefore very hard to spot. So you really do need methods that find things of very low abundance amongst species of very high abundance. And this is really an issue of dynamic range and being able to measure accurately inside what is often interpreted as just noise. So, of course, we know that there are a number of challenges facing sequence variant analysis. There's so much microheterogeneity in many of these samples that it might be ambiguous or difficult to see what's in there. And, of course, you might not be able to separate many of them chromatographically either. There might be a number of post-translational modifications that weren't anticipated or miscleavages and so on. The list is reasonably endless. And I have an example here from a quotation from one of the papers that highlights how even in a stretch of 11 amino acids, if you just change one of those, you might end up expanding that search space by a factor of 191. So it's really a mathematically huge problem if you don't approach it logically. The rise of tandem mass spectrometry is apparent to almost everyone who's worked in the field. And of course, it might come as a surprise to some of our younger audience to discover that, in fact, some of this work was being done way back in the 1940s. And really, by the time you get to the 1970s, you end up with instruments that are reasonably routine and commercialized. And in the case of one of the people who used to work in the organization that I work in, tandem quadrupoles have become the bread and butter of many a clinical trial and are extremely well deployed worldwide for quantitative analyses. Now, at the time, it was a much more revolutionary thing than it appears to be today, but essentially, it's still one of the best ways of looking at a targeted transition between MS and MS-MS, in other words, between precursors and fragments in a very specific manner. So it's a very targeted type of tool nowadays. Now, as we go through into the late 90s and we get to the early 2000s, we also have not just tandem mass spectrometry, we have tandem and hybrid. And here, of course, I refer to tandem in space rather than necessarily tandem in time. But what's critical here for the work that we've done today is the combination of being able to have accurate mass on both precursor and fragment ion information makes the sort of work that Jason does possible. And for industry, it really is that these can be used routinely now a decade after the bigger revolution that promoted them. And they get used in ways that is way beyond what was envisaged back then. So now as we go through the 2000s and after 2005, after a flood of data that came from mass spectrometry, people started to look at better ways of doing these experiments. So there are more techniques now than were available. And initially, of course, many of the techniques were targeted. In other words, you knew pretty much ahead of time what you were going to start looking at or doing it on the fly as you went along. And that gave you some very clean data, but in fact it also meant that each new hypothesis needed a new experiment and you had to redesign it every time. So when you get to the later part of the 2000s, you start to get more and more techniques that are data independent. In other words, realistically, what you're doing is acquiring everything at once and then data mining post-acquisition to see what exactly was in there. So you do have a tremendous advantage of being able to adjust your hypotheses and going back and looking to see if that is true or not. But it does require improved data analysis and informatics to be able to do that analysis. Now, we are all familiar also with how databases were used, and sequence variant analysis is also applicable here. So in the past, people would do an enzyme digest of their protein or their therapeutic of interest or of a number of proteins together, and you'd do a database search to see what you could match. And from those matches, you would build out a protein identity. And of course, there are variations too, such as error-tolerant searches. But the real problem lies in what I have here in the red box, is that there was a huge amount of manual data curation. Now, if you have a lot of cheap postdocs available in an academic setting, that might not be a tremendous problem, but in industry, it's very hard to cope with both the time and the expertise needed to do that sort of work. So as we advanced further along, the better way of doing it was then doing the acquisition without any preconception. And the way in which it can be done today is by generating taglets, and you look at the taglets and decide if they're hot or not. Now, of course, this is not really a disco term. It refers to whether that taglet has a lot of information associated with it or whether there is less information associated with that taglet. Ultimately, what that means is that you end up with a number of credible sequence variants in this case, but with evidence that you can investigate further. And hopefully that list will be a lot more sensible to curate and have a lot more evidence for how you've drawn up your hypothesis or what you've planned to match that against. Now, the very techniques of searching databases have changed as part of this. Historically, there was often a brute force mechanism where you said, well, I'm going to try all of these modifications inside the search space. So if you look at the white box that I have there, it's a sort of discrete part with the black box below it. And in the white box, you will try all of the modifications for that region of the database. And you would never try other modifications, such as those that I have in the black box in the database search. Now, this, of course, is not necessarily very satisfactory if you know that the reality is somewhat more complex than that. So that's where understanding that the search space is not monolithic comes in. And that's where the Paragon algorithm gets used to look at feature probabilities. So if you imagine that what I have there is a representation of my database from blue at the top to red at the bottom, getting different colors representing heat, if you like, I can try modifications for hot segments of the database. So everything, if you like. I can try only the most likely ones for the segments that are warm. And I can try only the most likely modifications for cold segments where I don't have a huge amount of information to rely on. And of course, that also applies to digestion features and tolerances and so on. So really, you're using a much more information-rich approach from the outset and not wasting resources in areas where you can't really fish out what's happening. So to take that on into industry and where it might be useful to perhaps do it all the way through, I can envisage that at the early part of the organization in discovery, You might be doing sequence variant analysis for a number of reasons, such as efficient clone selection, which is what Jason's doing, the security of your IP, failing candidates very early if they're going to be poor, and understanding codon changes. And you'll see some of that coming through. As you go through development, of course, you might then want to be testing what exactly are your cell lines doing with different harvest regimes or with different feed. But as you get towards production, you might then instead really just be monitoring that the variants that you made in the very beginning are still there or still aren't there, depending on whether they need to be there or not. So overall, this becomes something that's much more usable in the real world and much more usable for an industry that needs to gain its efficiency and actually needs to fill its pipeline, needs to know much more about its product, and also have a great deal of security about what it provides to its patients at the end. So it remains only for me to show you the obligatory legal disclaimer and to thank you very much for listening to me. But luckily, after me comes a much greater scientific brain, and I hope you enjoy listening to the thorough analysis of what he's done in the real world. And we, of course, would like to thank him very much indeed for pushing us very hard in the right direction.
All right, and that second speaker is Jason Hogan, a scientist at Bristol-Myers Squibb. Jason, before you get started, though, I just want to remind everyone about our Q&A session following the presentations and encourage everyone to submit questions through the Ask a Question box. All right, Jason, we're ready to begin.
Thank you very much, Tamlin. So we'll start with an overview and an introduction of sequence variants. We'll show a few case studies in today's webinar of amino acid mis-incorporations present in our expressed protein samples, and we'll demonstrate the use of Protein Pilot to assist in identifying amino acid mis-incorporations, and show a case study using MSMS to identify somatic mutations in an antibody variable region. When you look at biologics product development, we can divide the product development into four main categories, starting with exploratory discovery, going through early discovery, through full discovery, and finally exploratory development. Much of my work here at Bristol-Myers Squibb is focused on exploratory discovery and early discovery, where we typically focus on forming the target identification and lead discovery of our biologic therapeutic. When you look at where amino acid variants can be observed, starting in early discovery, we mostly focus on looking for the identification of DNA variants, leading to incorrect confirmation of the proper sequence of the target ID that we're looking for. As you move through full discovery and looking for lead therapeutic selection, you can also find amino acid sequence variants as a function of your production clone or growth conditions. And finally, through exploratory development in clinical trials, you're focusing on limiting any amino acid sequence variants for the manufacturing of your clinical product. So when you look at where origins of protein sequence variants can occur, we start at just the DNA level, and you can have a replication error on approximately 10 to the minus eighth per base, and that forms a separate DNA sequence variant. The other way to actually find where amino acid sequence variants that will occur as a function of amino acid mis-incorporation, this can be split apart to either from when the DNA is being translated or being transcribed into RNA, or it's being translated from the RNA into the protein sequence. Both of mistakes in either of these cases will form those amino acid sequence variants, which then form a final product of a protein sequence variant. We'll move to our first example, and we focus solely on peptide mapping for antibody batch comparisons. And this is a light chain peptide. And when comparing it, a prep scale batch versus a small scale batch, you see an extra peak in the HPLC chromatogram. And we use peptide mapping to identify each of these peaks. This peak of the prep scale, as being the correct peak, is shown at the top of the screen with the light chain peptide. And the small scale mass is 16 Daltons below the expected peptide mass. So when looking for amino acid sequence variants, that 16 Dalton shift can be identified to either glutamine to IRL, IRL to proline, or serine to alanine. We also use peptide mapping to look for any sort of chemical modifications that occur as a function of protein production, such as oxidation, deamidation, different glycosylation forms, and lysine clipping, and so on. So then you focus each of those peptides in that HPLC chromatogram to MSMS. The top scale, the bottom picture showing the proper peptide, and the top one showing the improper peptide with the amino acid sequence variant. And you see you get the same exact fragmentation spectrum in both cases, except for the difference between the Y4 and the Y5 ions, showing that in the bottom correctly formed peptide, there is a serine at the fifth to last position. And in the top picture, the improper formed amino acid variant was where the serine was exchanged for an alanine. MS-MS is pretty much the staple of identifying any of these sequence variants, but for much of the part, the intact mass really leads us in the right direction to which amino acids are possibly being mutated based on the mass shift. So we move to a second example, a case study, where we didn't know anything about the amino acid differences that occurred, and we also only had information from a single DNA case study, not having any sort of gold reference standard to compare any future mAb lots against. So in this case, we started with an antibody that was generated by hybridoma, and it is shown in the upper left. And the isothermal titration calorimetry experiment showed that it showed a 90% functional concentration. We had this mAb reformatted to an IgG1, and it is shown in the middle. And we also made a separate mAb shown in the upper right with also an IgG1 with an FC portion mutation. Now, having these ones straight out of production, the MAB2 IgG1 showed 90% functional concentration, but MAB1 IgG1 did not. Showing the KD values were similar for all of the MABs by Biacor shows that at least the binding efficiency is the same for each of the MABs. So when we move to the next slide, we'll show that the heavy and light chain of these two IgG1 antibodies, the light chain shows exactly as expected. The heavy chain, on the other hand, did show a 16 Dalton mass shift that was lower. Now, we have no reference standard available for this protein, but only the expected amino acid sequence. We typically do our heavy and light chain mapping in concert with peptide mapping, so as one piece of information will verify the other. So we'll go ahead and subject our antibody sample to our antibody analysis workflow, and it just starts by simply denaturing our antibody, reducing it, alkylating it. In this case, we used iodoacetamide. We do a trypsin enzyme digestion and subject the peptides to LC-MS-MS analysis, and we then search our database using Protein Pilot. We have two instruments in our capabilities in our laboratory. We have a 5600 triple TOF and an older LTQ Orbitrap XL. Our HPLC is a typical standard HPLC with a Waters UPLC BEH C18 column. And then for our database search, I always put in our protein sequence of interest, along with a larger database. In this case, I've used a yeast database in order to give the search algorithm more false answers to look at, which improves in identification statistics. So when you look at what the database search algorithm, Protein Pilot, we've chosen this one to help backstop our data analysis because it's a smarter processing algorithm for protein identification, as St. John alluded to in his talk. And it does a good extensive identification with their Paragon algorithm, allowing you to search for hundreds of biological modifications and unexpected cleavages without exploding the real search space and thus increasing your database search time. When looking at how the Paragon algorithm works, it is all based on generating these taglets. And so what it does is it's a smart search algorithm that analyzes your data looking for amino acid sequence tags. It then combines these sequence tags to form these taglets out of each MS-MS spectrum, and each taglet is then rated with a chance that the taglet is correct. So it is doing essentially a limited de novo search of your data, allowing you to then do a tag-based search against your sequence database, and thus to eliminate a lot of false answers so that you can then widen your search space to look for modifications. When we look at how the antibody analysis workflow works with these taglets, is it then ranks them all based on the number of degree of certainty, and then maps them to a specific protein, and this is referenced in the Small Cell Proteomics paper in 2007. And then each segment is given a sequence temperature value, and thus in this picture shown as the STV, where the more taglets you obtain, the higher the sequence temperature value. when we look at how the Paragon algorithm is based on using these taglets, it allows the search space to vary based on the sequence temperature value. So the more taglets that you match to a particular peptide sequence, the higher the sequence temperature value, and it is then searched more extensively for modifications. This follows the base assumption that it is worth more effort to find a molecular weight match with stuff with more tag evidence, indicating that the right answer may be found there, and then it will allow then softer digestion rules, it'll increase the number of modifications it will search, and larger, bigger molecular weight deltas. The goal of the Paragon algorithm is to score fewer peptides while keeping the right answers, which it does very well. And the value to the end user is it does create more identifications, better discrimination of search identifications, but a main focus is search speed. So in a simple example of a protein pilot search output, it is divided into 4 main sections. The top of the section of the original output window shows your individual protein identifications, and they are ranked based on the number of peptides that are identified at 95% or higher. If you move to the middle part, the middle left side of the screen, if you click on any particular identification in the top window, it then shows all of the grouped protein identifications that match that sequence as well. So it also helps pick out of your database protein families. The middle part on the right side shows all of the individual peptide identifications that are linked to that individual protein identification. And as you click through the list in the middle part of the left screen, then it will actually highlight all of the peptides that match that specific protein and blue out ones that came from other homologous protein identifications. The bottom of the window shows the sequence alignment, and it color codes things based on how likely at least the peptide is positively identified, with green being 95% or higher. In this example here, we see very, very good sequence coverage of this protein target, and this database was searched using a lot of germline database information, so there's a lot of homologous protein identifications. Now in the last slide, if you click on the left side of the screen looking at an individual peptide identification, which I've shown here in the top of this slide, then the bottom window shifts from being the protein identification overlap to the individual peptide database. And so this shows your MS-MS spectrum of that with the alignment and the sequence in the lower left portion of the screen. The benefit of this is it does all identified fragments are flagged in bold print in the left picture. And this can be toggled on and off by clicking on any individual M over Z value in the table, or you can remove all B or Y plus 2 ions just by clicking the entire column. You're able to also zoom in and out of the mass spectrum, and so this makes a very user-intuitive way to analyze your data in a visual architecture. It easily makes us a much faster approach to sort our data and then identify and confirm that many of our amino acid sequence variants are correct. So when we look back to our original example with our IgG MABs that one showed functional activity of only 50%, we went ahead and did our antibody sequence analysis, straight trypsin digestion, ran it through Protein Pilot, and we were able to first identify the incorrect peptide, which is known as the MS-MS spectrum in the bottom panel, and it identified a tyrosine to phenylalanine mutation straight out of the search results. And it shows the B3 and the B4 ion identifying that change of 16 Daltons from the expected protein sequence. Interestingly of note out of this experiment is that it also identified a large amount of the properly formed expected protein sequence, showing the tyrosine at that position. When we move this back to looking at the antibody peptide map with our UV signal, we have then able to confirm from the MS-MS spectra, without any prior knowledge, that this sample was contaminated with a DNA mutation. We then looked back at the UV signal, where we were able to find that the correct peptide resulted with 37% of the possible UV chromatogram absorbance, while the Y to F mutation peptide resulted in 63% of the absorbance. When we track this back to the original DNA sequence, the location of this tyrosine shows that it was an A to T mutation in the DNA sequence, which led to the basically chimera of these two products being created. When we go back to my overview slide looking at biologics product development and looking for all of these amino acid sequence variants, there's been a larger push to look for these amino acid sequence variants earlier in the product development. The potential payoff is this can provide downstream benefits by working on codon optimization, helping you with cell line clone development, and looking for expected hotspots for modifications. When we look at how these amino acid substitutions may occur, you can look at the RNA codon table. This one was downloaded from Wikipedia. And these sequence variants are most oftenly a result of a single base pair event to the expected amino acid. So we would expect them to be either located either vertically or horizontally in the RNA codon table from the correctly encoded amino acid. When we look at these observed sequence variants across our two MABs that we just showed in the previous example, the one MAB1, this is the IgG1, showing the 50% functional concentration. MAB2 is the other one, the IgG1, with the FC portion mutations. And we do see a small amount of mutations in each of these cases, and they're usually very, very low, under 0.3%. We have seen some as high as 1%, but typically you're around 0.1 to 0.2% of the mutations. Now, these modifications, shown in the most left column, have varied from anything from cysteines to tyrosines. A lot of them are glycines to either glutamic acid or aspartic acid. And one we see very common is serine to asparagine. We can trace these modifications back to where the mRNA codon mutations are occurring. And oftentimes you do see that a G to A mutation in either the second or even sometimes the first position can occur. We do much of our quantitation based on the ion chromatogram signal from the mass spectrometer, largely because these numbers are too low for any UV quantitation. When you take a look at how these mismatches can occur leading to your amino acid sequence variants, this was a great presentation created by Zhongqi Zhang from the American Society for Mass Spectrometry conference last summer in Minneapolis, and this talk can be downloaded from the ASMS archives. But I have summarized it here in the next few slides, showing that where he found that a GU mismatch occurs frequently in RNA structures, and the two areas that we can see, we can directly limit these GU mismatches to either a translational error or a transcriptional error. So when looking at the left side of the picture, a translational error is where the mRNA would show a G, but when the tRNA comes in to bind, instead of correctly using the C to G base pairing, it does that GU mismatch. And so the improper tRNA then puts the improper amino acid into your protein sequence instead of the correctly encoded serine. This can also be done on a transcriptional basis as the antisense DNA is being synthesized into messenger RNA, where a GU mismatch can occur instead of a proper CG pairing, and that leads to then the improper mRNA being transcribed, and then it does a proper tRNA to mRNA connection. So your protein sequence is then, in this case, a proline was converted to a serine due to the DNA to RNA mismatch. Zhongqi Zhang showed in his talk at ASMS that where he did some statistics on the types of mismatches that occurred, and found that the largest one was this G to A mutation in your mRNA to tRNA mismatch. He found another one occurred in the third base pair, where you would either get C or U converted to A or G. And then smaller mutations, our transcription errors, only accounted for about 10%, those shown in the previous slide, or U to C or U to A inversions where the RNA had problems with the tRNA. He showed that the CU, UU, and AG mismatches that occur in these RNA structures, they can occur but are less common. The third position is less stringent during the codon recognition, and we can see that any third position GU mismatches are typically self-correcting, as they typically encode the same amino acid, except for the cases where methionine goes to I or tryptophan is a stop codon. So when you look back at our sequence variants observed in our produced antibodies, where we have the GU mismatches, we can then link them back to the items out of Zhongqi Zhang's talk, showing the majority is our G of our messenger RNA going to the U of the tRNA. We did identify one transcription error and a few other of the rarer mismatches that he occurred. So the biggest concern we see from our amino acid case is that serine to asparagine one, which is typically a G to U mismatch. And we seem to find that with the highest percentage and more common across the samples. So when we look at where, using our database search algorithms, they do have pitfalls, and Protein Pilot is no exception, is that false positive identifications are always an issue, and this is worsened by the low abundance of the variants. So it is difficult to identify quality MS-MS spectrum, leading to the identifications and thus site-specific confirmation of where the amino acid sequence variant occurs. Each MS-MS spectrum must have an identification. And our scoring of functions, we use decoy databases to provide assistance, but these poor quality MS-MS spectra can still lead to high scoring peptide matches. And the onus then is on the user to determine the quality identifications from the poor ones. One benefit of using the newer high-resolution accurate mass instrumentation, in typical our case using the 5600 triple TOF, is this provides an added ability for us to filter the identifications for errors. So we look at a final example where we can use these high-resolution instruments to then identify where these mutations occur, largely because the accurate mass of the MS1 information gives you a hint as to where your modification is occurring, and then MS-MS is used to identify things. We have a case study here where we had an anti-idiotypic antibody clone that was no longer viable following storage, and it was lost. Unfortunately, we were not able to do any DNA sequencing for this antibody clone, and so our solution was to use mass spectrometry to identify all of the CDR regions, and so we can make a recombinant expression of this antibody and compare it to our original protein lot for a newly developed replacement clone. In this example, we will use Protein Pilot to identify any germline-specific somatic mutations in our remaining antibody material. This workflow has been adequately discussed in these following papers, two of which are by Genentech and one by Cell Signaling Technologies. In this case, instead of showing the data set that I can show today, we can show one that every user can go ahead and take a look at, and this involves the use of the Waters intact mass standard. In this case, our example is a protein A-purified mouse IgG1 antibody. The amino acid sequence is published, and the link is provided at the bottom of the slide. This contains germline mutations in both the light chain and the heavy chain variable regions, as well as the IgG1 domain. This protein sequence is pyroglutamic acid on the N-terminus and is C-terminal lysine clipped. So we will subject this to our antibody workflow that we showed earlier in this presentation, where we start with our antibody sample. Instead of doing just a single trypsin digestion, we will do a multi-enzyme digestion, subjecting them to multiple LC-MS-MS runs, and then searching the database using Protein Pilot. Our database here will be a custom germline database, and we'll start with that, and then using our standard workflows where as we identify more information, we then use our protein sequence of interest and compare it against our protein sequence that we believe is correct and a yeast database for any false identification statistics. So when looking at how our database search will work in the first pass, we need to start by taking our LC-MS-MS database and map it to these peptide spectra to a specific germline. So when we look at this, we make a custom database of all possible variable regions, D region, J regions, and since we know it is an IgG1, we'll just use that single conserved region. The database search program will start to link all of these peptide spectra to specific germlines, and as they can start to map together, a lead sequence will emerge with the most peptide hits. In this case, we've identified the variable region 1 in the upper left of the slide. As you start to map all of your peptide sequences to a projected possible entire sequence, you will get a lot of overlap across these different digestions, but you will have a few gaps where no sequence coverage is obtained. And this is where your somatic mutations will be present. So in our case of looking at this Waters intact mass standard, we start with the light chain of the Waters intact mass standard, and it identifies the immediate light chain to be correctly identified as the germline 1-117, and identifies the J5 region and a kappa light chain. In this case, Protein Pilot was able to identify 100% sequence coverage of this, and it identified only the single CDR1 mutation. Our germline shows in the bottom of the slide the CDR1 peptide where it has a S to Y mutation, and that is the only identified DNA mutation in the light chain. When we move over to the heavy chain of our intact mass standard from waters, we were able to identify the correct germline very easily as the 2-6-7 mouse germline. I have then put the heavy chain sequence in the middle of the 3-set slide, and our observed sequence will be identified and confirmed using Protein Pilot. Identifying of multiple DNA mutations requires us to use an iterative database search where we take information from one of the digests where only a single mutation is identified, then link that into a successive iterative database search, and then it identifies more mutations, which can then essentially bootstrap you to the complete sequence of the somatic mutations. In this case, we were able to identify all of the modifications leading to the variability of this antibody from the expected germline sequence. We were able to correct an identification here in the middle part of the slide. There is a serine to threonine mutation from the germline, but we were able to correctly locate it to a residue, 2 residues up to a serine that was not labeled in the waters sequence. We were also able to confirm an error in the labeling of the intact mass standard from waters. In the upper right on the first row, there is an MG, but we were able to confirm by MS-MS that it is actually matched the germline GM. So those cases, those peptides were positively identified, and only by interrogating the MS-MS spectra were you able to confirm that the amino acids were correctly matching the germline and not the published sequence. Now, when we look at the benefits of using Protein Pilot, its main benefit that I think goes unheralded is its use of graphical user interface and the ability to easily review your identifications and then easily click on and off fragments for easy use of viewing your data. The other benefit of Protein Pilot is that it is instrument vendor neutral, and it does accept the MGF file format. It can be created in both a 1-core and an 8-core software license. In our house, we use the 8-core software licenses, and that benefits of uses the ability to use multiple processors. So in summary of this webinar, we've shown that we can show amino acid sequence variants can be identified using high-resolution accurate mass instrumentation. And this accurate mass information typically will identify the presence of your amino acid sequence variants. We then use MS-MS to confirm the location of all of these modifications, and this is always aided with the ability to have spectra with high information content. We use our Protein Pilot search algorithm, which can help us identify as well as site localize any amino acid mis-incorporations. And in our final example, we were able to show any antibody somatic mutations could also be identified using both accurate mass information, MS-MS, and subsequent iterative database searches. I'd like to acknowledge my colleagues here at Bristol-Myers Squibb, Susan Wong, David Passmore, Indrani Chakraborty, Jennifer Giuliano-Holm, Sujata Singh, Carla Henning, and Shrikant Deshpande, and Mohan Srinivasan for their assistance with all of the identification of amino acid sequence variants and subsequent data shown in today's presentation. I'd also like to acknowledge our AB Science colleagues Justin Blethrow and Eric Johansson for the data of the Waters Intact Mass Standard, demonstrating that Protein Pilot can identify any DNA somatic mutations.
All right, thank you very much, Jason. Before we start the Q&A session, I just want to give a final call on the questions for our panelists. Please submit your questions now, and we'll answer as many as we can. I also want to let you know that a post-webinar survey will deploy shortly in the presentation manager. You might need to disable a pop-up blocker in order to see it. If you could take a few minutes and respond, we would be most grateful. Okay, we're ready to start our Q&A discussion. We have a lot of good questions already, and let's get started.
Okay, so to add a little variety to today's webinar, this is John Sterling, Editor-in-Chief, and I'll be picking up the Q&A as we go along here. I've got some very interesting questions. So, Jason, let's start with you. The first question is, I've already got Mascot Server running in my lab. Can I teach it to do this kind of searching?
Yes, a Mascot server as well. The newer versions do have an error-tolerant searching function, and there is a good paper by Vish Kata at Genentech showing how to implement those into your normal workflows. Any other search algorithm with error-tolerant searching capabilities can do this type of workflow as well.
Okay, and another question, Jason. Can we detect mutations in germ cells using mass spectroscopy?
Yes, and I would believe that this technology can be adapted to even looking at DNA sequence mutations, looking for, say, phenotypes across different patients, germ cells. I think the main backdrop in regards to this question is the ability to have either a protein sequence database to start with or an underlying DNA sequence in order to start with your mutations. But the technology is directly applicable.
Okay. Another question, Jason. Have the interactions with discovery changed as you have started to do this work?
We're finding we're doing a lot more work looking at these types of mutations earlier in our discovery phase, essentially looking for clones that may be used to go forward, although when they do end up going into production, oftentimes that material can be cycled back to us for these types of analyses.
And another question, Jason. Let me just see. You've got a number here. What are the biggest benefits you have seen in this work for your organization?
I think the biggest benefit has been to identify the quality of some of our products. We've adapted the same workflows to looking at any in-house produced antigens, original clone selections for lead antibodies. It's basically more as a selection tool to pick a product that can at least be compared in progress of lead selection. So it's more a quality control at this stage in the game for us. Okay.
And St. moving over to you, how many sequence variants are allowed in the final drug product?
Yeah, that's more a question where the regulators would come in. So I should make sure that the audience understands that I've got to couch my answer in terms of numerous different types of regulations that occur around the world. The easy answer is that generally sequence variants aren't allowed, and in fact this is highlighted in some of the regulations for biosimilars where it's not considered necessary. This is a matter of debate because, of course, there are sequence variants being discovered all the time in products where it wasn't known, perhaps in some cases. So things will change, perhaps, but in general, the main restriction on people producing biotherapeutics in general is that you have to know whether the sequence variants are there or not, and of course it's expected that they should be avoided. And that is probably the general rule around the world for most regulatory authorities. But for details on each one, I would highly recommend that you go and read those regulations for that specifically. And of course it's always going to be one of those things where the FDA says, well, you absolutely have to come to us early on in the process if there are sequence variants analysis, and they highly encourage that. So it's a regulatory aspect, I think.
And St. we have a very direct question here. Did you imagine you could have done this kind of work a few years ago?
So for a lot of people, this was extremely challenging, and certainly the cases of multiplying the amount of information to search through was huge. There are two things that have probably made this very much more achievable in recent years. One of those is having very fast MSMS. That's allowed the sequence of the peptides to be found more easily in general, and being able to do that. And the general intelligence going into informatics is the other one. So being able to, instead of treating databases as a monolithic whole, to actually understand what's going on as part of your detection system, and being able to find taglets, for example, was quite a leap forward, I think, in many of those cases where you know where to go and expend your effort to go and look for things. So a combination of things came together, I think, in the last few years as people have started to think about the applicability to the biotherapeutics market.
Thank you, St. Jason, turning back to you, how long does it take to do these partial de novo searches? Do you do this for all your clones now?
We typically take about anywhere from 30 to 90 minutes per database file, and we're now working this into all of our clones that are subjected to peptide mapping.
Okay. And Jason, another question. Do you know if the CDR hypervariable regions show a higher tendency for somatic mutations?
It seems to depend on the different germ lines that are used, but you do see a higher percentage of somatic mutations in the hypervariable CDR regions. We also do see, in cases like looking at the Waters intact mass standard, you also see these somatic mutations in even the conserved regions, which may be just differences in the different animals used.
Okay, and Jason, number 19. Please explain the benefit of including a decoy database in searches.
So the benefit of decoy databases in your searches is it gives your scoring algorithms more false hits to look at, and it allows you to refine a lot of your answers by giving it a more statistical probability of being correct. In our case, we use decoy databases commonly when doing more high. It's more applicable to people doing high throughput protein identification experiments. In our case, a better example of a decoy database is just tacking in our expressed protein sequence into a larger database like yeast or E. coli, something to give the search algorithm a lot of wrong answers to look at, which then when it filters them for statistical probability, then your true protein sequence climbs right to the top. It is typically avoidable to just do a single or very small protein database, as it does allow more false hits to filter through.
Okay, and we have a question for Eric Johansson, who will identify himself in a second. Eric, how about post-translational modification suggestions? Is it possible? Eric, are you there?
I'm here. Sorry about that. I was temporarily on mute. Yeah, I was just going to say that one of the interesting features of that partial de novo style searching that Jason was talking about is that it's equally useful for post-translational modifications as well. You search for as many as like 350 PTMs while searching for sequence variants at the same time, and it will have the same benefits of lower false positives and higher true positives.
Okay, Eric, thank you. And St. will accurate intact protein mass help to speed up the identification of sequence variants and localization of the variants?
John, that's quite an interesting question. I think a lot of people will assume that you can localize things at the intact level. Generally, this is regarded as impossible. The variations are so numerous that the ambiguity is quite high. So for most people, it's going to be an approach where you might find that there are differences at the intact level, and then you'd go and localize them using a peptide map. And I should also stress that having accurate math at the intact level probably plays almost no part in this. It's really at the peptide level and for the fragments where the accurate mass comes into play. At the intact level, you tend to have broad peaks, multiple overlapping heterogeneities that becomes increasingly difficult to determine what those variants are. So at the intact level, you're generally, as Jason showed, you're generally saying, oh yeah, there are differences in these ones, and then going on to localize them at the peptide level, and also getting the sequence provided at the same time to then determine exactly where in that peptide that change has occurred.
Thank you, St. And Jason, if you have a custom germline database, will you have a subsequent differentiation database for personalization? He's referring to slide 48.
Yes, starting with the custom germline database, you can use the initial first pass of your search results to identify the type of family that leads to the germline. From there, once you identify some specific mutations, we then make a differentiation database to use that as a template to base all future searches against, allowing you to then even localize even more somatic mutations.
Okay, and Eric, we have a question for you. It's a little complex, so bear with me. You may be aware of this introduction to protein pilot data analysis. It's University of Minnesota. I'm not quite sure what the question here says. It's a database search program, protein pilot. I guess this person is looking for some comment on that program, if you know that.
It looks like they're suggesting an academic review of protein pilot. I guess all I can say is thank you for pointing that out to me, and I'll take a look at it. Okay,
perfect. Well, unfortunately, it looks like that's our last answer, as our time is up. I want to thank everyone for listening to this webinar, and thanks also for the great questions. The webinar will be archived for 6 months on our website, and that's www.genengnews.com. If you miss parts of it, you can watch it again, and you can also recommend it to friends and colleagues, which we highly recommend. Once again, a big thank you to our panelists, and to our sponsor, AB SciEx. Bye-bye for now.